Welcome. So in this video we will learn all about the milk immunoglobulins. So let me first define immunoglobulins. So immunoglobulins are nothing but they are the glycoproteins which are produced by the blood plasma B cells in response to the antigens. So whenever antigen, maybe it may be bacteria, virus, fungi, chemicals or synthetic substances which then enter into our body, then plasma B cell will produce antibodies in order to counter the bacteria, viruses, fungi, or the synthetic substances in order to fight against these external substances. And the antibodies are produced by the plasma B cells. Now how they are coming into the milk? They are coming into the milk from the blood itself. So blood is the source of immunoglobulins in the milk from all the species. Now we see that mature cow milk contains 0.6 to 1 gram immunoglobulin per liter, but colostrum is having very high concentration. So colostrum of yours is having higher concentration of the immunoglobulins, that is 100 gram per liter, and the level decreases rapidly postpartum until it becomes normal to 0.6 to 1 gram immunoglobulin per liter. Now in there are 5 classes of immunoglobulins which are known, that is IgA, G, D, E, M, and among them only 3, that is IgA, G and M, are present in milk. So out of the 5 immunoglobulins which are D, E, M, A, G, only M, A, G is present in milk. Okay, now these occurs as subclasses also. So Ig exists in IgG1 and IgG2, and if somebody asks you that which is the major immunoglobulin which is present in cow milk, it is IgG1, and which is the major immunoglobulin which is present in human milk, it is IgA. Okay, so always remember that the major immunoglobulin which is present in cow milk is IgG1, and major immunoglobulin which is present in human milk, it is IgG. Now IgG consists of 2 heavy and 2 shorter polypeptide chain that are linked by disulfides. So we will see the structure that every immunoglobulin is having 2 long heavy chains and 2 short polypeptide chains which are linked by disulfide, and we will see the structure in the coming slide. Now IgA consists of 2 such units linked together by secretory component and a G component. So in IgA, what we have the 2 basic subunits will be there. That is, units means that 2 heavy chain, 2 light chain is 1 unit. So in IgA we will have the 8 chains, that is 2 subunits, and they are joined by the secretory component and the J component. So that also we will see in the structure. Now IgM is having 5 linked 4 chain units. That is to say that it is having 10 antigen binding sites, or in other words that it is having 8 such units. That is to say that it is having 10 chains it will have. Okay, so chain, if we will count for 2, it is 1, it is 4, so 5 for 5 it will be 20. So 20 chains will be there in case of, and 5 subunits will be there. So 5 subunits of the 4 chain units, so 4 into 5, 20 chains will be there in case of IgGM. Okay, all in detail we will study in the coming slide. So the heavy and light chains are specific to each type of immunoglobulin. So heavy and light chains, they are specific to each type of immunoglobulins, and that also we will see some aspect in the coming slide. Now here the slide shows the concentration of the immunoglobulins in colostrum and milk concentration. So if you see that immunoglobulins in cow colostrum and milk, and also in buffalo colostrum and milk, and then in human colostrum and milk. So if you see here, as I've told you, that IgG is the major immunoglobulin both in the colostrum of cow as well as in the colostrum of milk. Okay, now in human milk the major immunoglobulin is IgA, both in the colostrum as well as in the milk, that we can see here. So the slide shows the concentration of the immunoglobulin. So here we can see that among the cow, buffalo and human milk, the maximum concentration of immunoglobulin G, that is IgG, is there in the buffalo milk. So here it is higher than 57.2, and also in milk also. So we can say that concentration of colostrum is slightly higher in case of the buffalo colostrum as well as milk among the 3 species, that is cow, buffalo and human milk. So the concentration of the immunoglobulins in colostrum and milk is shown in gram per liter. It is shown in the slide. Now see the importance. What is the importance? So we know that when calf is born without immunoglobulin in its blood serum, and hence it's very susceptible to cancer. So calf is born without immunoglobulins. So we know that the calf is not having the immunoglobulins in its blood serum, hence it's very susceptible to infection. But the intestine of the calf is permeable to the large molecules for about 3 days postpartum. So 3 days postpartum it is permeable to the large molecules. This means it can take intact and active form from its mother colostrum. So it can take, because it is the intestine is permeable to the large molecules for about 3 days postpartum. This means that immunoglobulin can be absorbed intact and active form in its mother colostrum. Now immunoglobulins from colostrum appear in calf blood within 3 hours of suckling. So within 3 hours of suckling it will be there in the blood of the calf, and that persists for 3 months, although the calf is able to synthesize its own immunoglobulins within 2 weeks. Although it can synthesize its own immunoglobulin within 2 weeks, but it persists for 3 months. And of course it is then essential, it becomes essential that calf should receive colostrum within a few hours of birth, otherwise it probably die. So it is very important that the calf should receive the colostrum as a source of immunoglobulins, otherwise it will probably die, as calf is also not having is not born with the immunoglobulin in its blood serum. So it becomes essential to feed colostrum within a few hours of birth of the calf. Now human baby contains immunoglobulin in utero. So in uterus itself the human baby is getting the immunoglobulins, and hence unlike the calf it is not dependent on immunoglobulin from milk. It is not dependent on the immunoglobulin from milk. But immunoglobulin in human colostrum is beneficial to the baby as it reduces the risk of intestinal, that it increases the immunoglobulin. So although the baby is not dependent on the immunoglobulins, as these are being obtained in utero by the baby, but then it is essential or beneficial for the baby as it reduces the intestinal infection. Now if we see the structure of immunoglobulins, we will see here in the coming slides that these are the family of immunoglobulins have related structure. All the immunoglobulins have related structure, and that we will see that, and each immunoglobulin molecule, as I have told you, that it consists of 2 light chain, that is of 23 kilodalton, and 2 identical heavy chain of 53 to 75 kilodalton linked together with disulfide bond. So that we will see in the coming slide. So these heavy chains are there, light chains are there. These are linked by the disulfide bonds. A Y-shaped structure is formed. So we can say that a Y-shaped structure of dimer LH is formed. Now dimer or polymeric units of immunoglobulins. So if we have 2 such units, they are held together by a J component, that is joiner chain component, that is of 20 kilodalton protein chain. That also we will see in the coming slides. Now N-terminal region of the molecule specifically bind to the antigen. So antigen N-terminal bind the antigen, and the C-terminal has a effector function, that is it binds either the complements or the surface receptor proteins of the cells, and also if the antibody is there, that is if secondary antibody is there, then also that will be binding to the Fc region. So effector function include immunoglobulin binding host tissues to various cells of the immune system, merging some phagocytic cells with the first complement, and thereby causing the activating the immune system. So but mean to say that here in the structure, if we see, so this is the structure of immunoglobulin. So if we see this structure, it is having 2 heavy chains. So one heavy chain is this, 2 heavy chains, and also 2 light chains. So here one light chain is here, 2 light chain is there, and then it is having 2 antigen binding sites. Here we can say the 2 antigen binding site is there, and this site is known as the Fc region. So this fragment is known as, that is C stand for crystallizable region. So this is crystallizable, and also this is a region where the your complement will be binding, or your surface receptor proteins, that is surface receptors proteins on the cells, that is these cells receptors are called as Fc receptors, and thus activating the immune system. So they will activate the immune system for causing the further that is a series of reaction will take place to fight off the bacteria, viruses and fungi which is entering in the body. Now we have see, as I told you, that heavy chain that is consisting of one variable region. So this is one variable region of the heavy chain, and then here 3 constant regions are there. Here we can see the 3. Now we see here that 2 heavy chains are linked by disulfide bonds, and also light chain and this heavy chain are also linked by the disulfide bonds, that we can see. Now the carbohydrate is attached to the CH2. CH2 region of the heavy chains. That is the constant region of the heavy chain CH2, that the carbohydrate is attached, as can be seen from the slide. Now this region, that is the region between the CH1 and the CH2, is known as the hinge region, and this gives flexibility to this structure, and then thereby these two sites can act independently on the antigens. So flexibility is given by the hinge region, that they become flexible and they can independently handle the different antigens at their own. So flexibility is given by the hinge regions. Okay, so again repeating that two heavy chains, two light chains are there, and heavy chain is having one variable region, three constant region, whereas in light chain we have one variable region and one constant region. So variable region means that they're having a variable composition of the amino acid among different immunoglobulins within a species, whereas constant region means that they are having constant sequence of the amino acids among different immunoglobulins within a species, although some variations may occur among different species, but within a species they have constant sequence of amino acids, and that is why these are called as the constant regions. So here it is G1, that is it is having 4 chains, 2 heavy chain and 2 light chains, whereas in case of IgM there are 5 subunits, 1, 2 subunit, 3 subunit, 4 subunit, 5 subunits, and 5 subunits. So there are 5 subunits, and IgA 2 molecules of IgA are joined by J component or also the secretory component. Sometimes also we have the secretory component. J component is of 20 kilodalton around, and secretory component we see that is of around just I will tell you, that is secretory component molecular weight. If you see it is around, just to tell you, it is 79 kilodalton. It is of 79 kilodalton. Okay, so yeah. So two IgA component, that is 2 subunits are linked by J component and the secretory component. J component is having a 22 kilodalton of molecular weight, whereas the secretory component is having 79 kilodalton of the molecular weight. Okay, now as told you that antibody is having 2 light chains, which are either can be K or lambda. These are called as K, kappa, or the lambda. 2 heavy chain are of 5 types, that is can be mu, delta, gamma, epsilon, or alpha, and the type of heavy chain defines the class of immunoglobulins. So IgM will have the mu, D will have the delta, G will have the gamma, E will have the epsilon, and A will have the alpha heavy chains. Okay, so these are how they are called. That light chains are of 2 types, that is of kappa and gamma, lambda, and whereas heavy chains are of 5 types, that is mu, delta, gamma, epsilon, and epsilon. Now variable regions, I have told you that end regions and of the both chains are a variable region, that is amino acid sequences vary among the class of antibodies in a species. Of course it is varying among the species also, whereas in constant region we have the similar amino acid sequences within a class, each class of antibodies in the species, and although some variation may occur among the constant region of the different species. Okay, now let us see about the epitopes, paratopes, FAB region and FC regions in the structure of the immunoglobulin. So epitopes are the surface structure that is present on the antigen which binds to the antibody. So antigen is having some structure, some part of the antigen that is binding to the antibody, that is known as the epitopes, whereas the part of the antibody on which the antibody binds, that is known as the paratope. Okay, so the part on the antigen is called as epitope, whereas part on the antibody is called as the paratope. That antigens can easily bind on the specific site named paratope, which is located on the variable region of the antibody. Okay, now antigen binding region on the antibody and comprises of N-terminal variable region of the light and heavy chain. So here we can see that this is antibody fraction, that antibody fragment is there. So here we can the light chain and the heavy chain. That FAB is on the antibody comprises of N variable region of the light and heavy chain. So it covers both the N region of the heavy and the light chain. Now FC region folds from the C terminal ends of the 2 heavy chain, and PFC region, as told that is the crystal region, and then as which are having amino acid sequence which are specific to the species. As I've told you that these are specific to the species, but among species they can vary. Antibody, if some anti-antibody was there, for example in LFA techniques or in ELISA techniques, we require secondary antibody. Secondary antibody means the antibody which has been produced against some other antibody which is produced in a particular organisms. So this is secondary antibody. So secondary antibody also binds to the Fc region. So secondary region also bind to the Fc region, that is also known. So that it may be complement bind, it may there may be the Fc receptor of the cell which may bind to activate the immune system. So this happens only in the Fc region of the antibody. Now if you see the very important, the physicochemical properties of the major bovine immunoglobulin classes. So I told you that in bovine milk that we have IgG, IgA and IgM, and the sedimentation coefficient of IgG is 7s, it is 11s and 19s. So here the sedimentation coefficient is 7s, that is indicating that it's the molecule of low molecular weight, whereas IgM is having molecule of higher molecular weight, that is sedimentation constant is 7s, 11s and 90s. So just to tell you what do you mean by sedimentation constant. So sedimentation constant indicates how faster a molecule will move when subjected under centrifugation. So 7s will move slower and the 19s will move faster or sediment faster during centrifugation. That is to indicate what do you mean by sedimentation coefficient. Sedimentation coefficient is nothing but the rate of sedimentation. So if a particle which is having higher size will sediment at a faster rate, whereas it's obvious because if a particle is having higher size, it will sediment at a faster rate, whereas a particle which is having is of lower size will sediment at a lower rate. So 19s mean to say that IgM is having highest molecular weight, that is the 960 kilodalton among the three, so it is having higher sedimentation constant, whereas the IgG is having lower sedimentation constant, that is lower molecular weight, molecular mass, that is 150 kilodalton, so it is having lower sedimentation coefficient. Thank you. Now if you see the molecular mass, that is 150 and IgA is dimer, it is around 300. Of course it is having secretory component, J component also, so it is 385 kilodalton, and then IgM is having around 5 times of this, that is around 960 kilodalton is the molecular weight. If we see the number of basic 4 peptide units in this, we know the IgG is having 1, IgA is having 2 subunits, IgM is having 5 subunits. Now heavy chain is heavy chain of the IgG is called gamma, of A chain is called alpha, and M chain is called as the mu. Okay, so light chain can be a kappa or lambda. Similarly here it is also kappa or lambda, here it is also called the kappa or lambda. Now if you see the molecular formula, so it can be gamma to kappa 2 if the light chain is kappa, and then if the light chain is gamma, it is called as lambda, then it is called as gamma to the lambda 2. And depending on the valency, it will be alpha to kappa 2, alpha to kappa 2, because Ig can exist in dimer, so monomer to tetramer, so it can valency may vary to 2, 4, 6, 8. Okay, so the formula will be alpha 2, K2 and the valency. For in IgM we have 5 subunits, so formula will be mu2 K2 or mu2 lambda 2 depending on the type of the light chain. Now valency of antigen binding, so obviously it is 2 antigen binding sites are there in the if I would like to show, as I've showed you also, that there are 2 antigen binding sites in a IgG. One is this and second is this. Okay, one is this, second is this, so 2 antigen binding site is there. So in IgG we have 2 antigen binding site, in IgA we have 4 binding site, 1, 2, 3, 4, and in this we have 10 antigen binding site, 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10. We have 10 antigen binding sites. Okay, now carbohydrate content. As I told you that it is the CH2 region to which the carbohydrates are attached in the heavy chain immunoglobulins, and the carbohydrate content in IgG is around 3%, in IgA it is 8%, and IgM it is 12%. Now approximate percentage of the skin protein, if you see that IgG1 has 1.2 to 3.3 as IgG1, and IgG2 is 0.2 to 0.7 as IgG2, and of course IgG1 is in the highest concentration. In IgA, that is the percentage of skin protein is around how much? It is 0.1 to 0.7%. Similarly in IgM also around 0.1 to 0.7%. So now let us see some points about the immunoglobulin G, IgG. Now IgG consists of a single 4-chain molecule with a sedimentation constant of 7s with a molecular weight of 150 kilodalton, as we have seen in the table also. Now major subclass of this molecule IgG1 accounts for nearly 80% of the total whey protein of the cluster. This is accounting for the total whey protein, 80% of the total whey protein of the colostrum. This is IgG1. That is to say that IgG1 is the highest amount that is present. However, its level far sharply below in milk weight sharply. The concentration of above 100 mg per ml has been reported in bovine colostrum, even higher than 100 mg per ml that has been reported in the bovine colostrum. Now if we see the level of IgG1 to IgG2 is 15 to 20 times higher in bovine colostrum, while in milk IgG1 is 4 to 7 times greater than IgG2. So we can see the differentiation in the concentration of the Ig and G2, that is 15 to 20 times higher in bovine colostrum, whereas in milk it is 4 to 7 times greater than IgG2. Now IgG1 has more acidic amino acid in its H chain, unlike Ig2 which has more basic amino acid in the H chain with p values of 6.8 and 8.3. That is mean to say that IgG1 will have the more charge and Ig2 will have the least charge, and because of that IgG1 will move faster in the electrophoresis, gives faster electrophoretic mobility to IgG1. So the charge on IgG1 will be higher, which will cause IgG1 to move faster in the electrophoresis. Now if we see the biological function of immunoglobulins, the maternal immunoglobulin confers a high degree of immunity to the neonates. So if somebody asks what is the biological function of immunity, we know that maternal immunoglobulin G confers a high degree of the immunity to the neonates. How? In humans, IgG molecules or substances can pass the placental barrier, and that is to say in utero they obtain the antibody and confer high level of the passive immunity to the newborn in case of the human baby, whereas in case of bovine, placenta acts as barrier and passive immunity is nearly absent, requiring external support of antibodies, and that is why colostrum is found outside. So IgG is an antitoxin class and plays an important role in secondary immune response, which include complement fixation, that is, and also the phagocytosis of the microorganism by ingestion through Fc receptor. So biological function of IgG is to provide the passive immunity to the calf, and also it is giving the secondary immune response, which is the complement fixation and the phagocytosis, and also it is an antitoxin class. Now coming to secretory immunoglobulin, that is secretory IgA is a major immunoglobulin class of human milk. I told you that in human milk it is a higher concentration, whereas in bovine milk it is IgG1 which is in the higher concentration. Colostrum, unlike the bovine milk, IgA exists mainly in 180 dimeric form and has a molecular mass of 385 kilodalton. So that is, it is existing in a dimeric form, that is, and has a molecular mass of 385 kilodalton, and this 118 are the amino acid residues. Okay, so just mainly in the 180 dimeric form and has a molecular mass of 385 kilodalton. The protein is associated with a glycolytic secretory component. So I have told you that in the structure we have the secretory component which is having 79 kilodalton molecular weight, and also J chain which is having 20 kilodalton molecular weight, as we have seen in the in previously also. Now we see the biological function of that is secretory immunoglobulin A, that in addition to milk, secretory immunoglobulin A also occurs in the intestinal tract. Molecules give protection against invading pathogens. So if a pathogen is invading, then it will bind to their antigenic sites, thereby preventing their adherence to the epithelia, and then it protects the nursing infant from the gastrointestinal invasion and by pathogens, especially E. coli, causing diarrhea. So this is the major biological function of secretory immunoglobulin, that is it binding to the antigen, preventing their attachment or the adhesion to the epithelial cells of the intestine, and thereby preventing the diarrhea, especially against E. coli causing diarrhea. It protects nursing infants from the gastrointestinal invasion by pathogens, especially E. coli causing diarrhea. Now, if we see about the some points about the immunoglobulin M, they're very important immunoglobulin, very much of high significance for a direct technologist. It is having highest significance for a direct technologist. Now, IgM molecule is a pentamerase immunoglobulin with 4 basic chain structure that we have already seen. Now, individual heavy chains have a molecular mass of 65 kilodalton, while intact molecule has a molecular mass of 917 kilodalton. Now, level of carbohydrates in IgM is as high as 12%, obviously, because it is pentameric, so that it will be having the maximum concentration of the carbohydrates. And molecule also contain J chain, which binds the pentameric polypeptide chain structure. So J chain, I've seen they are having molecular weight of 20 kilodalton. Now, if you see the biological function of IgGM, they are most effective against the invading microorganism. It is the most effective, and if the first immunoglobulin to be secreted in response to antigen, if somebody antigen is entering, that the first immunoglobulin which is generating is the IgGM, and its production begins within 2 to 3 days after the antigen is first encountered. That within 2 to 3 days its production begins. And IgGM is extremely efficient agglutinizing and the cytolytic body, so it is involved in the agglutination as well as the cytolytic destruction, that is causing the cell to die, bacteria to die, fungi to die. That is by showing the agglutinizing as well as the cytolytic activity. Now, if we see that significance of IgM, as I have told you, that it's the immunoglobulin which is of the greatest interest to the technologist. Why? Because it promotes the clustering of fat globules leading to enhanced screening. How? So because IgGM is having different antigenic sites which binds to the different epitopes of the fat globules, and thereby causing the networking, increasing the size of the fat globule, may cause also aggregation. And then since the size of the fat globules increases, this means that velocity will increase. That is to say that velocity is directly proportional to the diameter, or the directly proportional to the r square. So more is the radius, so velocity of rise will be increased. That is to say that creaming will increase, and that hence it is involved in the cold agglutination of the fat globules. So this is the which resulted in the cold agglutination of the fat globules. So the IgGM is the most significant to the dairy technologist. So this is all about the basic about the milk immunoglobulins. Thank you.